Do you have your assignment on DNA replication? Have you accomplished this assignment? About this? What I'm going to explain is on how the DNA replicate or divide. You know the structure of the DNA. And you know also that the DNA, the oxyribonucleic acid, is double-stranded. It is double-stranded. And last time, I also explained to you the components of the DNA and the messenger RNA. I explained to you the components of the DNA and the messenger RNA. I also explained to you about complementary base pairing. And then I explained to you how the nucleotide polymerize, or how the addition of the nucleotide occur. I also explained to you the importance or significance of the link or the bond that join the components of the DNA. Michaela Alabado? Michaela, are you in?
Yes, po. Yes, Doc.
Can you give me the three components of the nucleotide or the DNA? What are the three components of the nucleotide?
Nucleotides po are composed of one sugar molecule, a phosphate group, and nitrogen-containing base.
Nitrogenous base, nitrogen base.
Yes, po.
Are you familiar with this nitrogenous base? The different types of nitrogenous base? Are you familiar with this? Michaela?
Yes, po.
And what are
The
they?
purines and pyrimidines, Doc.
Oh, what? What are the types of purines? What are the types of pyrimidines?
Purines po are adenine and guanine, and then pyrimidines are cytosine, thymine, and uracil po for RNA.
For RNA. That's
Yes,
right.
po.
Jera, are you in Jera? Ira or Jera? Arango, are you in? Jera. Absent?
Sir, si Jaira po yung unstable connection and si Miss Yoli po.
Unstable connection. Okay lang yon. Basta wag lang siya maging unstable, ha? Okay lang yan, maging unstable ang connection. Wag lang siyang maging unstable. Okay. How about you, Angel? Angel. Are you present, Angel?
Yes po, Doc. Yes, Doc. Angelo po.
Angel, you are teaching at FEU.
Yes po, Doc.
As you can see, it is open. It is open, and then it is replicated. And component or the structure that is responsible for the replication of the DNA, is the DNA polymerase, both on the leading strand and the lagging strand. Okay, you have to see the leading strand in the diagram. Sora, can you hear me? Sora,
Yes, okay.
can you hear me? Can you see the diagram? Sora?
Yes.
Okay. Sora, this is the DNA, the oxyribonucleic acid. By the way, are you familiar with DNA, Sora? Are you familiar with DNA?
Yes.
Okay. So, you look at this one. You look at the leading strand. You look at the leading strand. This one is the leading strand. And this is the double-stranded DNA. In order that the DNA will be replicated, the first thing that will happen is that the DNA should be relaxed. It has to be relaxed. Well, what I mean by relaxed, you know, the DNA is tightly coiled. It is tightly coiled and it has to be relaxed. In order that the DNA will be relaxed, it needs an enzyme. And the enzyme needed for the relaxation of the DNA is known as topoisomerase. Okay. Once the DNA is relaxed, it will be opened up. Okay. In this case now, the DNA is open. Okay. And the enzyme that is responsible for the opening of the DNA, is known as DNA helicase. It's known as the DNA helicase. This is the one. The DNA helicase, it opened the DNA strand and then have it separated into two single strands. Okay. So, once it is open, replication occurs. But, okay, it is being replicated. The leading strand is one. It is continuously replicated by the DNA polymerase, as you can see. It is continuously replicated by the DNA polymerase. Okay? Whereas the lagging strand, it is not continuously replicated. The replication process is discontinuous, as you will see. But before the lagging strand will be replicated, a set of helix destabilizing protein will bind into the lagging strand so that it will be straightened. Okay, it will be straight because the lagging strand will not be replicated if it is not straight. Okay? And what makes it straight? It is the helix destabilizing protein. Once it is, once it is straightened, okay, a primer, an RNA, a short RNA primer will be synthesized. A short RNA primer will be synthesized. And what is responsible for the synthesis of the RNA primer? The enzyme known as primase. This is the one. It is the enzyme primase that will synthesize the short RNA primer, as you can see. Okay? So, what will happen? Now, Okazaki fragment will start here. Okay. So what will happen, class, is that the DNA polymerase now will bind, it will bind into the primer, the short primer, and then it will start synthesizing the lagging strand. It will start replicating the lagging strand. Okay. So this is supposed to be replicated as in the case. Okay. So this is another, and you know, this fragment, when this will be synthesized into a new lagging strand, you will call that the Okazaki fragment. Okay, let's go to this one. Okay, here is the RNA primer. Here is the RNA primer. Then the DNA polymerase bind into this RNA primer, and it start synthesizing a new DNA strand. Okay, as you can see, it starts to synthesize a new DNA strand, the red one, from the primer, from the RNA primer. Okay, this is the new lagging strand, and you call this the Okazaki fragment. You call that the Okazaki fragment. In the next slide, class, this you will see how it works. Okay? This is the leading strand. In the presence of the DNA polymerase, what happened to this leading strand? It is replicated to a new leading strand. Okay, a new leading strand is synthesized continuously. But how about the lagging strand? The lagging strand, it has to be stabilized. And now the RNA primer will be synthesized by the enzyme primase. This is now the RNA primer. And then what will happen? The DNA polymerase will bind into this RNA primer, and then will start synthesizing the new lagging strand from this template strand. So this is now the new lagging strand synthesized by the DNA polymerase. And what do you call this new lagging strand? This is known as the Okazaki fragment. Okay? This is known as the Okazaki fragment. Then after that, it will stop. The replication of the lagging will stop. And then another RNA primer will be synthesized. And then the DNA polymerase will bind into the RNA primer, and then it will again synthesize a new DNA lagging strand. And this is the reason why the lagging strand is discontinuously synthesized. Unlike with the leading strand, which is continuously synthesized by the DNA polymerase. So, this case, you will see the difference in the replication of the leading strand and the lagging strand. Okay, did you understand the class? With the replication of the leading strand and the lagging strand. Okay, now, this is to show you how the lagging strand is replicated and how the Okazaki fragment is formed. Okay. Okay, this is the lagging strand. This is the lagging strand of the DNA. Okay? And this is the RNA primer. And this is the RNA primer. Okay. So this is how the lagging strand is replicated discontinuously. First, there is a primer. Okay? There is a primer. This is the primer. And then the DNA polymerase will bind. And then it will synthesize a new lagging strand from the old lagging strand. So this is the new lagging strand. Okay. And then after that, what will happen? The DNA polymerase will again synthesize. And what do you call this one? You call this now the Okazaki fragment. And then again, another primer will be synthesized. And then the Okazaki fragment will bind into the RNA primer. And then it will synthesize the new Okazaki fragment again, or the new DNA fragment, known as the Okazaki fragment. So, in this case now, class, as you can see, that there are two fragments that are newly synthesized, and these fragments are known as the Okazaki fragment. And what will happen to this RNA primer? This RNA primer will be removed. Okay? This will be removed. Okay? And this is what happened now. So, this will happen. And these two DNA strand is about to join. But there is what is known as a nick. Itong dalawang, these two Okazaki fragments will be linked or joined. And what is responsible for joining the two Okazaki fragments? What is responsible for joining the two Okazaki fragments? That is another enzyme will be involved. Another enzyme will be involved. And what is that enzyme? What is that enzyme? Anyone?
Ligate.
DNA
Ligase.
ligase po.
That is the DNA ligase. Okay? The DNA ligase. So, this is now complete. This is now the new lagging strand. But this time now, this new lagging strand will become again another leading strand when this will be replicated. Magiging leading strand na naman to siya. And the other one will become a lagging strand. What will happen to the primer? The primer will be removed. Okay? That is how DNA is replicated. Do you have any question? May mga tanong ba kayo? Did you understand how the Okazaki fragment is synthesized? Do you have that in your assignment? Joyce Reyes? Ever? Joyce Reyes, are you in? Have good knowledge about DNA replication. And now we'll go to DNA transcription. How the DNA is transcribed. Okay. Transcription and translation. Transcription of the DNA and translation of the messenger RNA. This is the central dogma. Okay? This is the central dogma of the transcription and translation. But actually, this is what is known as protein synthesis. How the protein is synthesized. Okay? What is the central dogma? That the DNA is transcribed to messenger RNA, and the messenger RNA is translated into protein. That is all. That is just what you will understand. Tandaan lang nyo ito, ang central dogma, transcription and translation. Just remember the central dogma. In the transcription process now, how does it differ from the replication of the DNA? How does transcription of the DNA, when we are talking of transcription, that means that the DNA is being copied. Okay. When we are talking of replication, that means the DNA divides. Okay, that divides. Transcription is the generation of RNAs from genes. What do you mean by generation? Synthesis of RNAs from DNA. It is the process of creating mRNA in the nucleus from the DNA of our chromosomes. This is straightforward, class. This is a straightforward statement of transcription. It is catalyzed by an enzyme called RNA polymerase, which copies the sequence of a DNA strand into RNA. This is straightforward statements. Okay? Remember, class, that in the transcription process, the enzyme involved is called RNA polymerase. Whereas in the replication, DNA replication, the enzyme that is involved is DNA polymerase. Okay? And this is the RNA polymerase. There are three types of RNA polymerase in the eukaryotes. One is known as RNA polymerase 1, which synthesizes ribosomal RNA. RNA polymerase 2, which synthesizes messenger RNA, and RNA polymerase 3, which synthesizes transfer RNA. So these are the three kinds of RNA polymerase 1. But the most commonly used polymerase that I will be discussing with you is the RNA polymerase 2, which is responsible in the translation process, wherein messenger RNA is synthesized. So, okay. So the RNA that transcribe, the type of RNA that transcribe the DNA into messenger RNA is the RNA polymerase 2. Okay, we will not discuss about the sigma factor because this is for the prokaryotes. This is for the bacteria. The three RNAs, what are the function? The messenger RNA, the function, the messenger RNA, it carries the genetic information and then translate it to synthesize protein molecules. Okay. The genetic information and then translate it to synthesize protein molecules. Whereas it also carries the coding information to ribosomes. And usually, class, the genetic information is encoded in the sequence of nucleotides arranged into codons, 3 bases. It is a code. The genetic information is a code. And it is made up of codons. And you call that the genetic code. That is why, class, what your assignment again is next meeting, I want you a copy of the genetic code. Try to look at the copy of a genetic code in the biochemistry book, microbiology book, or molecular biology book, or biology book. Okay? And the code is called is made up of codons. Okay? And it is consisting of 3 bases. But actually, class, it is nucleotides. It is nucleotides. But here, what you can see in the codons are the bases, are the 3 bases: adenine, guanine, cytosine, uracil, but not thymine. You know why you could not see thymine in the code? Why you cannot see thymine in the code? Maria Salome? Salome, how come you could not see thymine in the code? Salome, are you present? Salome, are you present? Gem?
Yes po.
Taladhay? Taladhay?
Yes, Doc.
Yeah.
Because
Why is it that you could not see in the codon the base thymine?
instead of thymine, uracil is found in the mRNA.
Why? How come you could not see the codon, the thymine in the codon?
Because when during the transcription phase, the while the transcription phase is ongoing, there is no coding for thymine in the mRNA strand.
Okay. The answer is because it is an mRNA. Huh? You could not see the thymine in the codon because it is an mRNA. And what can you see in the codon instead of thymine? Gem.
Uracil, sir, instead of
Uracil.
thymine.
What you could see is uracil. So, what the nitrogenous bases that you could see in the codons, they are only uracil, cytosine, guanine, and adenine, except thymine. Always remember that in mRNA, it is uracil that is found, that you could find. Okay? And that is the function of the mRNA. Ribosomal RNA. What is the function of ribosomal RNA? It is a large RNase that make up a substantial portion of the structure of the ribosomes. Okay? Central component of the ribosomes protein factory. Whereas the transfer RNA, and again, it is in the ribosomal RNA where the messenger RNA is bound. The transfer RNA class is able to match genetic information and shuttle amino acids across a cell. Okay. The transfer RNA. From the cell, okay, from the cell, across the cell, the transfer RNA is the one that carries the amino acid, carrier of amino acid. Carrier of amino acid. Okay? So the transfer RNA is a carrier of amino acid. It contains sequence of 3 nucleotide bases at one end called anticodon. 3 nucleotide bases at one end called anticodon. Always 3. The anticodon and the codon, there are only 3 nucleotide bases. Always remember that. We know that the opposite of the anticodon is codon. And where can you find the codon? You can find that, you could find the codon in the messenger RNA. Whereas the anticodon, it could be found at the end, at one end of the transfer RNA. So, the anticodon is being carried by the transfer RNA, and the codon is being carried by the messenger RNA. Always remember that, class. Prior to transcription, there is also localized unwinding of the chromosome or the DNA, which will allow the RNA polymerase to access the DNA. Unwinding of the chromosome in such a way that will allow the RNA polymerase to bind into the DNA. So this is now the DNA, the double-stranded DNA. Okay? As what I told you, again, as what I told you during the replication of the DNA, that prior to the transcription process, prior to the binding of the RNA polymerase into the DNA, it has to be unwind. Okay? The DNA, double-stranded DNA, shall be unwind. And what is that enzyme responsible for the unwinding of the DNA? It is known as topoisomerase. Again, it is known as topoisomerase. Okay? As you can see, that it is the DNA is unwind prior to transcription. Either strand of the DNA may be used as the template strand to be transcribed. Okay? Either the two could be transcribed of the DNA. Either this one or this one could be used as template strand for transcription process. As you can see now, here, the RNA polymerase bind into the DNA. And what do you call this region in here of the DNA? What are the steps in transcription? Initiation. Initiation usually takes place at the promoter region. That is the start, the initial start of the transcription process. It usually takes place at the promoter region. This is the key feature for control of gene expression. Okay? When we are talking of gene expression in here, it has something to do with the translation process into protein. The DNA, it is expressed into a protein molecule, and that is gene expression. And the promoter region, what is the importance or significance of the promoter region? It is the binding site of the RNA polymerase. In other words, it is where the RNA polymerase is bound prior to the transcription process. And what do you call the promoter region? What do you call the promoter region? In the prokaryotes, you call that the PreMRNA. Prokaryotes, bacteria. In the eukaryotes, you call that the Tata box. You know, this is the sequence. This is the DNA sequence. Okay? Thymine, adenine, thymine, adenine, adenine, thymine. You call that the PreMRNA box in the bacteria. But in the eukaryotes, you call that Tata box. Thymine, adenine, thymine. Or cytosine, adenine, adenine, thymine box. Or the GC box. Guanine, cytosine box. You call that the Tata box. And you call that the promoter region. And then the coding sequence contain coding information. The coding sequence of the DNA, that is the coding sequence of the DNA, it contains the coding information. And then there's also the terminator sequence, similar to the terminator sequence. There's also the terminator sequence, which is specified at the end of the mRNA transcript, or at the end of the DNA sequence. That is the terminator sequence. That means that when the RNA polymerase reaches the terminator sequence, transcription will stop. The RNA polymerase usually uses the DNA strands to make RNA copy of the gene. Okay? So these are the transcript. It is used, the RNA polymerase. And it is bound into the promoter region. This is the promoter region. The promoter region of the DNA, and this is the termination sequence. The termination sequence of the DNA. And this is how So what will happen is that the RNA polymerase will bind into the DNA, and then it will start the transcription process. Okay? It will start the transcription process. From okay, this is how. So the RNA polymerase, it moves from 3' to 5' direction, 3' to 5' direction. What happened? A transcript is synthesized. An mRNA transcript is synthesized. So this is how it happens. Okay, class? So as the RNA polymerase move forward, the mRNA transcript is synthesized. Okay. Okay. And this is what we call elongation. This is another step, elongation of the messenger RNA. The RNA is synthesized in a 5', 3' direction. And transcription start point, AUG. So this is now, as you can see, as the RNA polymerase move forward, there is an elongation of the messenger RNA. And then when the RNA polymerase reaches the termination sequence, the transcription will stop. And here is now the DNA. Okay? The DNA. Okay? The DNA is transcribed to pre-messenger RNA. And there are fragments in the messenger RNA that is coding and non-coding. And these are fragments of the intron. The intron is the non-coding portion of the messenger RNA. So it is being so this pre-mRNA has to be processed. At the both ends of the pre-mRNA, it has a cup. It has a cup. It has a 5 cup and it has a polyethyl cup. Polyethyl. You know the meaning of polyethyl. That means there are several adenine. That's why it is called polyethyl. There are several adenines. Okay. This is the pre-mRNA. Now, what will happen? The RNA, the pre-mRNA is being processed. And what happened to the intron? The intron is removed. Okay? The intron is removed. When the intron is removed, the mRNA became shortened, and this is now the protein coding sequence. This is now the one that is being translated into protein. The one that is being translated into protein. Later on, you will know how introns are removed and what is the significance of this cup, the polyethyl, ethyl, and the cup. And this is now the polypeptide or the protein. Okay, the transcript is processed into mature mRNA before it is exported to the cytoplasm. Understand, class, that translation of the messenger RNA occur in the cytoplasm. The 3 components for processing the pre-mRNA. That is the 5 cup or the RNA 7-methyl guanosine cup. So that is the one. And what is the significance or importance of this cup? Of this RNA 7-methyl guanosine cup. Its presence is critical for recognition by the ribosome during the translation process. Okay? That RNA 7-methyl guanosine cup is very important for recognition by the ribosome during the process of translation. Gem, are you in? Are you still in, Gem?
Sir,
Huh? Are you still in?
nawala po yung connection ni Ma'am Jamela.
Okay. So what will you do? Who is talking?
Noriza po.
Huh?
Noriza, sir.
Marina?
Noriza po.
Norina. Is that your first name?
Yes, sir. Yes, Doc.
Norisa.
Yes po.
Norisa. allow. Did you understand this, Norisa,
Yes po.
about the 5-metal cap?
Yes po.
So that the ribosome will recognize the messenger RNA. So that the 5-metal RNA 7-metal guanosine cap is very important for the recognition of the messenger RNA by their ribosome. Norisa, are you married, Norisa?
No, Doc.
You're single. May I know what is your
Yes po.
RNA metal guanosine cap so that you will be recognized by your boyfriend? What is your 7-metal guanosine cap so that you will be recognized by your boyfriend?
Wala pa po.
Wala ka pang boyfriend. How about you, Salome? Maria Salome Santos. Are you present, Maria Salome? Ano? Joyce. Joyce.
Yes, Doc. Sorry.
Ever? Reyes? How are you going? How are you? How will you? How are you recognized by your boyfriend? What is your metal guanosine cup?
I don't have a boyfriend po, Doc.
Okay, you start having a boyfriend so that. How about you, Raquel Flores? Raquel, are you present?
Yes, Doc.
Okay, Raquel. Do you have a boyfriend?
I'm married, Doc.
You're married. Okay. What is your metal guanosine cup in order that you will be recognized by your husband?
Doc, I think my height.
Your height. Okay. Your height. Okay. So that is the 7-metal guanosine cup in order that you will be recognized by your husband. Siguro malayo ka pa lang among the crowd, the crowded places, you could be easily recognized by your husband because you are a 6-footer. Raquel, you are a 6-footer?
No, Doc. It's the other way around. I'm smaller, Doc.
You're
But my husband is a 6-footer, Doc.
six
That's why.
footer.
Yes,
Okay,
Doc.
that's good. So you could be easily recognized by your husband even in a crowded places because of his height, isn't it?
Yes, Doc. Opo.
Okay, that's right. So now you should understand why ribosome could recognize the messenger RNA because it contains the RNA 7-methyl guanosine cup. Okay. Another thing also, it also serves as protection from 5 exonucleases degradation. In other words, class, in order that it could not be destroyed by these exonucleases, it has the messenger RNA as a cup. It is like a shield, a protection from 5' exonucleases degradation. Okay, did you understand me, class? Did you understand me? Now, the importance of this cup. Another is the polyethyl. What is the importance of this polyethyl? It stabilizes the mRNA. Huh? It stabilizes the mRNA. So, Jem, what should be done with their connections in order that it will become stable? Jem,
More hydrogen bonds po.
huh? What? So that their connection will be stable, what are you going to do to their connection?
Additional of hydrogen bonds po.
Besides that, what else? They have to place what? They have to place polyethyl cup in their connection so that it will become stable. Huh? Their connection wiring is like a messenger RNA. So in order that it will become stable, you place a polyethyl cup. Jem? And also it will prevent ribonuclease degradation. It will also prevent. So the 5-methyl 7-guanosine cup and the polyethyl cup, it has somewhat similar function. It protects the messenger RNA from further degradation of these endonucleases. These are enzymes that could cut the RNA. Okay, splicing. How introns are spliced is the process by which PreMRNA is modified to remove certain stretches of non-coding sequences called introns. Okay, how the introns are removed or spliced. Usually it is performed by an RNA protein complex called the SNRNPs, or small nuclear ribonucleic proteins. Okay, so these are enzymes class. This is small nuclear ribonucleic proteins. These are actually enzymes. What will happen? This will form a loop in the intron to form what is known as spliceosome. Okay? Spliceosome. And then it will remove the intron. This is the intron. It will remove the intron. And this is now the messenger RNA, the matured messenger RNA. And these are this is the intron, and this is the SMRNPs. Okay. And what do you think is responsible for joining the nick or the gap? There will be a gap here. There will be a gap. What is responsible for that? For joining the nick, for repairing the gap? Anybody? Michaela Alabado, do you know what is that? How it is joined, the gap? Michaela,
Yes
are
po.
you still in?
Yes po.
Do you know how that gap is joined? This one, magkakaroon ng gap niyan. Huh? How it will be joined? Do you know? Huh? Angelo? Angel,
Yes, Doc.
do you know what is that? What is responsible for joining, repairing the gap?
Wait lang, Doc. Nag-choppy po. Wait lang po.
Huh? What? What? Anyone? Ano bang ginagamit?
Ligase po.
Who is talking?
Salaw po.
Norisa.
Doc ligase?
Of course, ligase, Norisa. How come it took you almost 1 hour to answer the question? Talaga bang nag-iisip ka, Norisa? You are really a deep thinker. Talagang pinag-iisipan mo talagang pagsagot, kaya it will take you an hour before you could answer the question. Where did you encounter this ligase? Where did you encounter, Norisa? Where did you encounter this ligase?
DNA replication, Doc.
In the DNA replication. That is when the two Okazaki fragments were joined. You use ligase. Always remember that enzyme ligase is always used for joining DNA or mRNA fragment or strand. So that is how intron is removed. It is removed by these RNPs. This is small nucleic ribosomal protein, ribonucleic protein. Okay, so it will be now the RNA polymerase shall reach the termination sequence. And so it released the mRNA transcript from the DNA template. Okay, this is the difference when it comes to transcription translation. Transcription translation of the DNA between prokaryotic and eukaryotic cell. Okay? As you can see now, because there is no nucleus, there is no nuclear membrane, so transcription of the prokaryotic cell occur in the cytoplasm. Whereas the transcription of the in the eukaryotic cell occur inside the nucleus. Inside the nucleus. And translation occurs in the cytoplasm. Here, both transcription and translation in the prokaryotic cell occurs in the cytoplasm. Okay, so we have finished about transcription. Now we go to the translation of messenger RNA. By the way, Sora, are you still in? Sora,
Oh, yes.
okay, do you have any question about the transcription of the DNA? Do you have any question? Okay. So we go. Yes, Sora.
I need to student it myself after class.
Okay. So if you have any question anytime, you could ask me.
Okay.
Okay. So now we are going to discuss about translation of the messenger RNA. Translation is the process where ribosomes synthesize protein using the mRNA transcript. In other words, class, the messenger RNA transcript binds into the ribosomal RNA, where it will be translated into protein. So, translation involves changing the information from the nucleic acid to polypeptides. Okay. Translation involves the changing of information from the nucleic acid to polypeptides, from ribonucleic acid to polypeptide. That is the translation. Changing the information from messenger, from ribonucleic acid, RNA, to protein. And what are the key players? The messenger RNA, the transfer RNA, ribosomal RNA, and the amino acids. These are the key players in the translation process. Okay, coding regions. The coding regions class, okay, this is the coding region of the messenger RNA. The coding region of the messenger RNA from this one to this one. Okay, are composed of codons which are translated into one protein by ribosomes. So these are the codons. AUG, that is one codon. UUC, that is another. CUU. So the coding regions are composed of several codons. It begins with the start codon, this one, which is AUG. Okay. Start codon, AUG. Okay, and ends with the stop codon. These are the stop codon, UAA, UAG, or UGA. Okay, so this is the stop codon, UGA. So translation will stop when it reaches the stop codon, UGA. The start codon is the starting point for translation. AUG is the starting point for translation. A stop codon, that is the termination of protein synthesis. That is the codon responsible for the termination of protein synthesis. Each codon encodes for a specific amino acid, except the stop codon. In other words, class, all these codons are coding for an amino acid. Okay? Like for example, AUG is coding for methionine, amino acid methionine. UUC is coding for amino acid phenylalanine. Okay? So, in this case, class, the codons are made up of 3 nucleotide bases. That is why, class, you bring the genetic code. Next meeting, that will be your assignment. Bring the genetic code and then learn how to interpret the genetic code. How this codon represent what amino acid does this codon represent. Okay? And sometimes the codons are coded several times, meaning that an amino acid is coded several times. Okay? An amino acid is coded several times. So steps in translation. The mRNA enters the cytoplasm and becomes associated with the ribosome, specifically the ribosomal RNA. That is the start of the information it encodes. The initiator tRNAs, it contains the anticodons, which is the opposite of the codon. And this pair up with the mRNA codons inside the ribosomes. Okay? Like for example, AUG is codon. And what is the anticodon of AUG? Anyone? What is the anticodon of AUG? Anyone? The anticodon is the opposite of codon.
Hello po, Doc.
Yes? What?
UAC,
Who is this?
Noriza po.
Noris?
Norisa po.
Norisa. Oh, did you understand, class? Ah? UAC. That is the anticodon. Of course, it is complementary base pairing. Okay. Complementary. The anticodon is the complementary of codon. Okay. So, there are three important components of the translation process. One is the messenger RNA, which binds into the small subunits of the ribosomal RNA. And the mRNA is the one that contains the codon. And the other one is the transfer RNA, which carry the amino acid, and it contains the anticodon. Okay, at the lower end. This is the transfer RNA. And the other one is the large subunit of the ribosomal RNA. And this is where the transfer RNA is bound. And what are the binding site of this large ribosomal subunit? They have the A site, meaning this the amino acid site. And then the P site, this is the P site where it is bound, where this transfer RNA is bound, the P site, or this is the peptide site or the peptidyl site, and the A site, which is the exit site. The exit site. So what will happen is that the transfer RNA will jump, the incoming transfer RNA will jump from the A site going to the P site and then going to the exit site. And then it is released from the ribosome. That is what happened when there is when there is protein synthesis. Okay? So, what will happen? What really will happen? Okay? These are the initiation factor 2, initiation factor 1, which will bind into the ribosomal RNA. Without the IF2, the initiation factor 1 and 2 translation will not occur. And also the initiation factor 3. Okay? They are these three factors that are very important for the translation process of the ribosomal RNA. Okay? As you can see in here, that there is the binding site in the large subunit of the ribosomal RNA. The A site or the amino acid site, the peptidyl site or the peptide site, and then the exit site. This methionine transfer RNA, this transfer RNA carrying the methionine, it all started binding into the A site. And then what will happen? It transferred to the P site. And then when it transferred to the E site, this amino acid will be left behind with another transfer RNA. Okay? This will be left behind with another transfer RNA carrying another amino acid. And so it will go from here to the exit site, and then it will be released. And then after that again, it will be looking at other amino acid where it will or it can carry. The elongation class mediates the entry of a transfer RNA carrying an amino acid into the A site. Okay, elongation. Here is the arginine. Okay, here is the transfer RNA carrying the amino acid arginine. This will bind into the A site. Okay? And here is now the transfer RNA. Nandito siya yan, ngayon lumipat dito sa P site, peptidyl site, where the start of polymerization, of forming a polymer. Okay? And this is the transfer RNA carrying the amino acid methionine. What will happen to this arginine? Okay? The arginine now binds into the A site. Okay? It binds into the A site. Where is the transfer RNA that is carrying the methionine? Okay. So it's there. Okay? Okay? You look at this class now. Arginine now is positioned, this transfer RNA now is in the A site. And this one is in the P site. And this transfer RNA came from the A site. Tapos lumipat siya sa P site now. And so what will happen when this transfer RNA will go to the exit site, and this transfer RNA will go to the P site? Okay, so this is what happened. This is what happened now. So when this go to the E site from the P site, what happened to the amino acid methionine? It is left behind. And then when this transfer RNA carrying arginine move from A to P site now, the methionine now binds to the arginine, okay, and it is forming, it starts to form a chain of amino acids, and this is what we call dipeptide. Okay, it starts to elongate by forming a chain starting with arginine and methionine. Okay, so what will happen to this transfer RNA? This transfer RNA will be released. Okay, so there is another alanine incoming transfer RNA carrying an amino acid alanine. Okay, and about to bind into the E site. Okay, so this is what happened. So now this transfer RNA is released from the exit site. Previously, this transfer RNA is carrying the amino acid methionine. Amino acid methionine. What will happen to this transfer RNA? This transfer RNA will move to the exit site, and this will left. These two amino acids will be left behind, and this will be transferred to, this will attach or bind to alanine. Okay, alanine, when this transfer RNA will move to the P site. Okay, so this is what happened. So in other words, all of these codons have been translated into protein. Okay, it has been translated into protein. AUG was translated to methionine. UUC, phenylalanine. CUU, leucine. AGU, serine. GGU, glycine. AGA, arginine. GCU, alanine. And GUA, valine. So, what will happen to this, again, to this transfer RNA? This transfer RNA will again move to the exit. And this polypeptide chain will be left behind. It will be left behind. So here comes another transfer RNA carrying an amino acid alanine. So it binds here, okay, and then it transfers to the P site. the one that is carrying alanine, and this one carrying the polypeptide chain is released. And this is now the polypeptide chain. And so, from here, this is the end of the translation process. Because the next codon is the stop codon. What will happen in the stop codon? A protein will bind into the stop codon so that there will be no more incoming transfer RNA. So translation will stop. Translation will stop. Then what will happen? What will happen? Anong mangyayari ngayon? What will happen then? Then after that the translation is stopped, all the components will be released. The polypeptide will be released. The transfer RNA carrying the polypeptide, it will also be released. The large ribosomal RNA will be released together with the small ribosomal, and then the mRNA transcript and the stop protein will also be released. So that is the end of the translation process. And then this is now the polypeptide. Where do you think this polypeptide will go after it has been synthesized? Where do you think it will go? Anyone? Where do you think this polypeptide will go? Michaela?
To the endoplasmic reticulum, Doc.
Okay, it will go to the endoplasmic reticulum, where it will be again processed. And then it will end up into where, Michaela? From the endoplasmic reticulum, it will be transported where?
To the Golgi apparatus po.
Then after that,
it will be released po, it will be released po in the transport vesicle po to the cytoplasm.
then it will go where? This protein will be transported where?
Outside the cell.
But before that, outside the cell, saan muna siya pupunta?
Cytoplasm po.
Ha?
Cytoplasm, Doc.
Of course, the cytoplasm. Going where?
Plasma membrane.
The plasma membrane, Michaela, it will, that transport vesicle you are referring, will fuse into the plasma membrane, then it will release the protein, and this protein will be distributed to all parts of the body where it is needed. Huh? Michaela. Then in the endoplasmic reticulum, it will be processed. It will go to the Golgi apparatus again, further processing will occur. So it will undergo further processing before assuming the three-dimensional configuration necessary for function. So processing will be done in the endoplasmic Golgi apparatus. Okay. Okay, that's it. This is how proteins are synthesized in our body. Okay, and this is happening now in our body. It is happening in our body. Because without this molecular process, our body will be non-functional. Our body will not have an enzyme to break down, to digest. Okay, do you have any question, class, about transcription and translation? Based on your assignment? Do you have any question? So next meeting, class, I'm going to discuss with you about defense mechanism, DNA defense mechanism. Bumalik na ba yung iba? Ha? Yes, you are. You are just listening. May mga connection na ba yon? You are still, you are. All of you are present. The 17 of you. 16. The 16 of you. Jem, may complete listing ka na ba? Do you have a complete listing?
Yes po, Doc.
Jem?
Yes po.
Mga ilan na bang estudyante natin talaga? Ang exact number of students? How many?
Including Miss Yun po.
Sura.
Yes po, 17 po.
17 talaga. Yes, in my list also 17. Wala nang additional 17. Sura, are you still in?
Yes.
Yes. Do you know Gemella?
No.
Gemella?
Yes, sir.
Gemella, you have to introduce yourself to Sora.
Yes, sir. I will
is
send
our,
an
is
I will send
our,
her an email po later.
okay. Gemella Magaluna is our coordinator, our class coordinator. And so, she can assist you or help you whatever you think, whatever you need in this class. Whatever you need in the class, you just contact Gemella, and Gemella could help or assist you. Huh, Sora?
Okay, thank you.
Okay, that's good. And do not, Sora, do not be ashamed of asking question. If you are some doubts, if you want some clarification, please ask question. Do not hesitate to ask question. Do not
Okay.
hesitate to ask help from your classmates. Okay? Okay, Sora? Okay, do you have any question, class? So I think I'm going to send you your reports. I'm going to send you your reports, and you just look at the Google Classroom.
Sir, I have
Your-
a question po.
Jem?
Can we have a copy po of your
Yes,
PowerPoint?
you can have a copy. You can have a copy in the Google Classroom.
Okay, thank you po.
Okay. So see you again next meeting, and have a nice weekend.
Thank you po, Doc.
Thank you, Doc.
Okay.
Thank you, Doc.
Thank you, Doc.
Thanks to Doc. Salamat.
Thank you, Doc.